Hello, good morning, students. So, we are done with the Unit 1 and Unit 2, and from today, we'll start with the next unit, that is Unit 3. So, Unit 3, the first topic is body fluids and blood. So now we'll see what are the subtopics that we'll have to cover in the body fluids. So contents are body fluids, then we have compositions of the body fluid and functions of blood, which is one of the very important topic. Then we have a hemopoiesis, formation of hemoglobin. Then in short we'll have to study about anemia. In brief, you all are going to study about the anemia in your next semester. Then we have a mechanism of coagulation. Mechanism of coagulation, which is a very important part as far as exams are concerned, and we have also already covered about this in our practical, that is bleeding time and clotting time. So how this clotting happens, what are the factors that are responsible for clotting, coagulation, we'll be studying in this mechanism of coagulation. Then we have a blood grouping. Then what is Rh factors, that will be also covered. Transfusion, its significance. Then we have a erythroblastosis. Then we have one disease, that is HTN, that is hemolytic disease of newborn. And then we have a disorders of blood, which will be covered in your subsequent semester. And the last subtopic that will be covered is reticuloendothelial system. So now we'll quickly see what are body fluids. So body fluids, they are the substances, or it is a fluid that is present inside the body of living organism. So it will serve as a medium for carrying the nutrients and waste product from the cells, and it also acts as a medium for carrying out the chemical communication. So it usually consists of water and dissolved solutes. So we have a solvent and solute. So these body fluids present in two compartments, that is inside the cell and outside the cell. So 2/3 of this body fluid is present in the intracellular fluid, that is in the cytosol, the fluid within the cell, that we have already covered when we did the cell. That is, we have a plasma membrane that separates the intracellular fluid from extracellular fluid. So plasma membrane separates ICF from ECF. So we had a cytosol in the intracellular fluid, and in that there were various organelles, and 1/3 of the extracellular fluid, that is ECF, which is present outside the cell. So it includes all other body fluid other than which is present in the cytosol, or you can say which is present in the cell. So the composition of extracellular fluid is 80% of the interstitial fluid, and 20% is blood plasma. So if we see the ratio of intracellular fluid and extracellular fluid, 55% of the total body water is present in the inside of the cell, that is the cytosol of the cell, and 45% of the extracellular fluid is outside the cell. Next is body fluid compartment. So this extracellular fluid, how it is been distributed in the body. So first we have a interstitial fluid, that is we have a 80% of the extracellular fluid is interstitial fluid, that is present between the cells. You have a various cells. You have various cells in the body. So whatever fluid is found in this region is interstitial fluid, and what fluid you found inside the cell is ICF. So then we have a plasma which is present in the blood, which is approximately 20% of ECF. Then we have a fluid which is found in the bones. Then fluid in the dense connective tissue, that is fluid is present in the dense connective tissue, that is cartilage. And then we have some other fluids like transcellular fluid, which include intraocular fluid. So now intraocular means the fluid which is found in the area of the eye. Then we have a digestive juices. So all the juices, or you can see the fluid which is found in the digestive area, what you can so it will include gastric juices. Then we have a fluid which is found in the esophagus. So all the fluid which include the GIT. Then fluid in the urinary tract, cerebrospinal fluid. So this will be covered. The cerebrospinal fluid will be covered in the when we do the CNS. So it is again a fluid which is a viscous, yellow in color, thick. Then we have a synovial joint fluid which we covered yesterday, and last is serous fluid. So this body fluid compartment is divided into two, that we have 45% of solid and 55% is liquid, that is in female, and in male we have 60% of the fluid. So the ratio of fluid in male is higher as compared to that of the female. So how this fluid is distributed is, it is divided into ICF and then ECF. So ICF will be the fluid that is in the cytosol, and extracellular fluid will include all the fluid which is found in the spaces, that is between the cells, all the fluid, and other fluids like one that is found in the plasma of the blood. Then we have cerebrospinal fluids. So composition of body fluid. So in body fluid we have different substances which include organic substances and inorganic substances. So in organic we have glucose, then we have amino acids, fatty acids, then we have hormones, enzymes, lipids and protein. If we consider inorganic one, we have cations and anions. So we know sodium cation, potassium, calcium, magnesium. Then anions we have chloride, phosphate and sulfate. So if we take the all cations or anions, the composition of cations and anion. So the most abundant cation which is found in the body fluid is sodium. So it is usually found in the extracellular fluid. So this sodium cation which is present in extracellular fluid plays very important role in muscle contraction that we have seen in case of neuromuscular junction, the sliding filament mechanism. Then it helps in the transmission of impulse from brain. And the last, which is also very important function of sodium, is fluid and electrolyte balance. So it helps in maintaining the fluid electrolyte balance of our body. So this will be covered in your urinary subtopic when you all are going to study the function of kidney, or how these desorptions of the sodium take place, and what are its adverse as well as what are the causes. Increase in sodium causes what type of defects, or decrease in sodium level causes what type of disease, will be covered in your next semester. So next anion which is present in a extracellular fluid is the chlorine. So it regulates the osmotic pressure. We'll see what is osmotic pressure is subsequently. Then it forms the main component of hydrochloric acid, that is it combines with the HCl, that is hydrogen in the gastric acid, to form the hydrochloric acid which is required in the GIT, especially in the stomach, to maintain the acidic environment in the stomach. Next we have most abundant cation in the ICF is potassium. So this potassium help in the maintaining the membrane potential at the resting position. Then we have it helps in action potential generation which happens during repolarization phase. And in case of sodium we have depolarization. Then it maintains the intracellular volume and regulates the pH. And lastly we have anions which are protein and phosphates. So next will be concentrations of body fluids. So concentrations of body fluid is usually expressed in three ways, that is osmolality, then we have osmolarity and tonicity. This is very important. So what is osmolality? It measures of fluid's capability to create osmotic pressure, which is called as the osmolality or osmotic concentration of the solution. So it is nothing but number of particles which are dissolved in kg of solvent. So how many solute particles which are dissolved in, that is osmol, how much amount of osmol which are dissolved in kg of solvent. Next we have sorry. Next we have osmolarity, which is another term to express the osmotic concentration. So in this case it is number of particle, that is osmol, per liter of solution. So it is number of osmol per liter of solution, that is liter, whereas in osmolality it is in kg. So remember this difference. Here it is osmol per kg of solvent, whereas in case of osmolarity it is osmol per liter of solution. The last term is tonicity, which is measure of effective osmolality. So we'll now see what is tonicity of solution is. So according to the concentration of solute, that is osmol, present in the solution, they can be classified as isotonic solution. Then we have a hypotonic solution, and third one is hypertonic solution. So you all must be now familiar with these two terms, that is hypotonic and hypertonic. Hyper means greater, that is something which increases. Hypo means less, or you can say it decreases. So isotonic is we'll see in the diagram. So isotonic solution has the same effective osmolarity as a body fluid, whereas hypotonic solution, it has the less effective osmolarity than the body fluid. Hypotonic has greater effective osmolarity than the body fluids. So now if we keep red blood cells, usually this concept is always explained in by taking the help of red blood cells. So now in hypertonic solution, the concentration of solute is more in the ECF. You can say, okay, you can see in this diagram that the solute particles are more in the solution as compared to that in the cell. So the fluid water H2O, you can see the fluid contents, will go from the RBCs into the outside environment. So it has to maintain the equilibrium. Whereas in case of isotonic solution, there is a equal distribution, that is solute particle outside in the solution are same as compared to the cell. So the equilibrium is constant in normal red blood cell. So they remain the shape of the RBC is same. Whereas in case of hypertonic solution, the concentration of solutes in the cell is more. As you can see the difference, there are very few solute particles in the solution. There are very few solute particles in the solution, whereas the concentration of solute in the cell is more. So the fluid will try to come into the cell, which will cause cell to swell. So it will increase the size because more amount of fluid is entering into the cell, which will cause bursting of the cell. Whereas in case of hypertonic solution, water is going out of the cell, so it will shrink, or you can say it will shrink, or you can say crenated. So the size decreases, or you can say it shrinks or shrunk in case of hypertonic solution, because the concentration of solute in the solution is more as compared to that in the cell. Whereas in case of hypotonic, the concentration of solute in the fluid is less as compared to that in the cell. So more fluid, water will try to enter the cell and it will cause the cell to swell. Whereas in case of isotonic solution, the concentration of water and solute is constant, or you can see it is in equilibrium. Next concept is significance of body fluids. So significance of body fluid is homeostasis, first one. So body cells survive in the fluid medium, which is called as a internal environment. So water not only forms a major constituent of internal environment, but it plays a very important role in homeostasis. Next important function is transport mechanism. So now body fluid forms the transport medium. So it brings nutrients from GIT, or from the site from where the nutrients are absorbed, and other essential substances which are required for our body enters the cell, and whatever unwanted or waste products are there, they are also being thrown out of the cell or body through the fluid. So it acts as a transport system. Next is metabolic reaction. Water inside the cell form the medium for various metabolic reactions which are necessary for growth and functional activity of cell. So metabolic reactions, whatever food or maybe drug that you all take has to undergo metabolism. So that process is also being influenced, or you can enhance by water body fluids. Then we have a texture of tissue. So it water inside the cell ensures the cell texture. Next is temperature regulation. So water present inside the cell, it also plays a very important role in maintaining the normal body temperature. So moving on to a main topic, that is blood. So whatever we covered so far, they were just a component which are required. So whatever we covered so far, they were just the components or introduction to body fluid. Now we'll come to the main topic, that is blood and its component. So the blood, which is as you all already aware, it is a fluid connective tissue. So the whole blood is divided into two components. So we have a first component, that is blood plasma, as you can see over here, and there are Pharm element. So this Pharm element consists of RBCs, that is red blood cells, then we have white blood cells, and then we have a platelet. So if you take a small sample of blood and if you centrifuge it in a test tube, then the top layer, what you can see, the hay colored liquid, is a plasma. Then we have a buffy coat, which include WBCs and platelets, and the last red color lesion which settles down at the bottom of the test tube are red blood cells. So this plasma which forms the top layer of the test tube, because it is less dense. So less dense, then we have the very viscous dense layer. It settles down at the bottom of the test tube. So if you take the whole blood, it is divided into blood plasma and Pharm element. So in blood plasma we have a water, major amount in the blood plasma is the water. Then we have proteins and other solute. So if we take the proteins, we have albumins, globulins, fibrinogens, and there are other substances dissolved in that. Then we have other solute which includes electrolytes, nutrients, then we have gases, regulatory substances, and some waste product. Then we have Pharm elements. Now Pharm elements include what? WBCs and RBCs and platelets. So in WBCs we have various different types of WBC, which include neutrophils. Then we have lymphocytes, monocytes, eosinophils, and basophils. Now these lymphocytes are again divided into T and B lymphocyte, T and B lymphocytes. And then we have a red blood cells, which gives a red color to the blood. So now we'll see the functions and properties of blood. The first important functions is it acts as a transport system. Now how it acts as a transport system, you already know that it carries the oxygen from the lungs, that is inhale oxygen from the lung to cells of the body, and carbon dioxide from the body cells to lungs for exhalation. So it carries oxygen and carbon dioxide is exhaled to the blood. It also carries nutrients from GIT. So whatever food that we take in, the absorbed nutrient from the esophageal or from all the parts of the GIT are being carried to the other cells through transport system, that is blood. So gastrointestinal tract to body cell, and also hormones from the endocrine gland to other body cells. So whatever hormones which are secreted by the glands, they have been also carried to their specific locations to the blood. Next is blood also transport heat and waste product to various organs for eliminations from the body. So now oxygens, nutrients have been taken. So whatever waste that is being generated is also been carried outside the cells or outside the cells to blood. It also acts as a heat transport system that we have already covered in the skin. So how whenever there is a very hot environment, so it will blood vessels will try to escape this heat from the body to the skin. Next function is regulation. So it helps in maintaining the homeostasis of all body fluid. It also regulates the pH through use of solutions like buffer. It also adjusts the body temperature through the heat absorbing and coolant properties of the water. Next important function is protection. So how it protects is that whenever there is an injury, whenever there is injury and if the blood starts flowing, it helps by the blood stops or the flow of a blood is decreased by forming a clot, that is coagulations, which we will be doing in subsequent lectures, how it forms and how the excess blood flow from our body is reduced. So which also protects against excessive loss from CVS system. So excessive loss from our body can be prevented by forming a clot. So various clotting factors are there which carry out this process of coagulation. White blood cells protect against disease by carrying out phagocytosis. So whenever there is injury, these white blood cells will carry over there and will carry out the further spread of infections, or they will carry out the phagocytosis of the microorganism whoever is trying to enter into the injured area and trying to carry out further infections. So that will be prevented by these white blood cells. Then the characteristics of the blood. So blood is dense and more viscous than water. So temperature is 38 degrees Celsius. The color of the blood varies depending upon its oxygen content. So if the oxygen is more, then it is very bright, and if the contents of oxygens are less, then it is darker in color. So the blood volume is 5 to 6 liters in male, in average size adult male, and it is 4 to 5 liters in adult female. So functions of blood elements. First we have a red blood cells, that is also called as the erythrocytes. So RBCs or erythrocytes are one and the same. So we have already seen it transport oxygen from lungs to body cells and carbon dioxide from body cells to lungs. Then we have WBCs, which are also called as leukocytes, or WBCs, which are also called as leukocytes. So there are several types of WBC. We have neutrophils, which we have seen in the diagram, eosinophils, basophils, monocyte, and lymphocyte. So each of these WBCs has its own functions. In lymphocytes, they are again of two types, that is T lymphocytes and B lymphocytes and natural killer cell. So this T lymphocyte usually helps in the immune response. T lymphocytes help in the cell mediated immunity, and B lymphocytes, they help in the antibody mediated reaction. And leukocytes protect the body from invalid pathogens and other foreign substances. And last we have platelets. So they don't have any nucleus, so release chemicals that promote blood clotting when vessels are damaged. So it will release some chemicals that will also help in promoting the blood clotting. So we are done with the basics or a very small topic of body fluids. In next class, we take how these all blood components are formed or clotting. Then we have formations of RBCs, that is erythropoiesis, will be covered in the subsequent lecture. Thank you.